Wonderful. Well, last but not at all least, we're going to be hearing from Dr. Denita Hadziabdic at the University of Tennessee, and she's going to be talking about thousand cankers disease, which is something that we haven't seen yet in Kentucky. I'm hoping that we don't see, but she's been dealing with for a while in Tennessee and can give us some updates on that. So she's an assistant professor at the University of Tennessee who focuses on diseases of trees. She has a PhD in plant, soils, and insects from the University of Tennessee, and her research focuses on understanding host pathogen vector interactions with a focus on thousand cankers disease complex. In addition, her expertise on tree health issues in our area, she is a US Fulbright scholar to Ghana, working on totally different systems and different things. So clearly has a really wide breadth of expertise there. So we're excited to have you joining today. Thanks for talking with us today, Denita.
Thank you so much. And thank you for the invite. You're having no issues hearing me and seeing the full presentation?
No, we can see both of those.
Wonderful.
And I'm going to spotlight your presentation. And just a reminder, if you have any questions for any of our past presenters, please put those in the Q&A and they'll try to get to them for you.
Thank you, and thank you for inviting me. And I'm very excited to talk to you about thousand canker disease and the research update for the past 10 years or so. And we dealt with this starting in 2010, basically. So what I'm going to be presenting today is a variety of research related to thousand canker disease, starting everything from the basic biology to trying to figure out a little bit better identification, because the pathogen is very slow growing. The system is very complex. We went everything from population genetics to microbiome, and now diving a little bit more into genomics. But before we go into any of that, I would like to, for those of you that are not familiar with thousand canker disease, so all walnuts are susceptible to thousand canker disease. Some are more susceptible than others, and black walnut is among walnuts the most susceptible species, where Arizona walnut is the least susceptible. And that one is in the native range of walnut twig beetle, which is the insect vector. And over the years, we found that actually that was not the only insect vector that potentially transmits this pretty devastating disease. Also, the fungal pathogen called the Geosmithia morbida is mainly responsible for thousand canker disease. What it does, it creates, I'm not sure, I'm just going to use my cursor here. It creates these cankers, which we believe they're annual cankers, and then the beetles basically have to create entrance and exit holes, and then they create these tunnels inside of the phloem area. And this is the white part is really mycelium, the fungal mycelium that is being spread throughout the tree. So as the beetles tunnel through the tree, they create these cankers, and these cankers can oftentimes coalesce, creating larger necrotic areas and eventually killing the tree in a very, very short period of time, roughly 3 to 5 years after initial symptoms are detected. So this basically at Colorado State University, you can see upper canopy die off, yellowing, wilting of the leaves. Part of the problem is also symptoms are very similar to any drought or sometimes environmental stressors, nutrient deficiency. So it is very hard to detect. So this is something that we rarely see in the Eastern United States. And we also see differences between East and West, which sparked a lot of interesting questions and led our group to do some neat stuff regarding biological control. So this is how Geosmithia looks under the microscope. And I like to explain, this is when you get pure culture, which is very difficult for us in the East compared to the Western isolates. And they basically look almost like pill-shaped, and they're oftentimes in these chains. So when the cankers coalesce, they create these larger necrotic areas. But for you to know that the disease is present, you have to peel the bark, which is usually oftentimes too late to detect the disease. This is the walnut twig beetle, Periopterus juglandis, a super tiny insect. And I have to do the full disclaimer for my entomology people. I'm on a fungal and plant pathology side of the equation here. And I work closely with several people from Purdue and University of Tennessee on the entomology side of the equation. So basically, this is the beetle under the microscope and these cankers that coalesce and create necrotic regions. So ultimately, why do we care? But TCD could become a global problem. It was initially detected in the US in the late 1990s and early 2000. The beetle was present there, but the pathogen was really identified only in 2011. And then magically from the Western US, it appeared in Tennessee. That was the first introduction to the Eastern United States. And since then, it has spread to several Eastern states. And in 2013, it has spread to Italy. So this is the only non-US basically country that has TCD confirmed. And as I said, all species of walnut are susceptible. And the reason we are very concerned because they're native and the preservation of biodiversity in native species is very important to our programs. So the TCD is not federally regulated. It is a state-issued quarantine in certain states in order to call that TCD positive state, which are all marked in purple. They have to have oftentimes both beetle and the pathogen present. In case of Indiana, for instance, walnut twig beetle, I think they found one, but they really found the weevils for Indiana, and we did find both beetle and the pathogen. And some other states found alternative vectors. So this is where the state regulation is a little bit interesting, depending from state to state. So what we noticed is that the TCD, and I'm going to refer for about 2,000 canker disease as TCD from now on. So TCD severity in the introduced range, which is Western United States, is very different. Oftentimes disease symptoms, you can see the arrows here, are very obvious, prominent. Upper canopy is dying, and in a short period of time, the tree is dead. Resulting basically in complete mortality in a short period of time. So what we're seeing in the Eastern United States are two different outcomes. So we're seeing that, for instance, this was the tree in Tennessee in 2010, very traditional, typical symptoms of TCD. And then this tree in 2014 seemed to be recovered to some extent. And I'm going to use quotes for that recovered, because there was a lot of epicormic shoots, and we do not know necessarily if they resprouted to a certain extent, but susceptibility and the host resistance is not there anymore. Or in the same general vicinity, another tree that was infected was completely dead 4 years later. So we're seeing these two different outcomes. And one of the first things we wanted to do is to develop these microsatellite loci, which are species-specific markers for identification of both beetle and the pathogen. So what that really means is basically we were able to multiplex, meaning we can have a multiple reaction in a single tube. We were able to confirm that here. So this is one marker, this is marker number 2, 3, and 4. We were trying to basically play with different concentrations and different combinations, because if we have multiple markers, we can have possible confirmation if they're working for 3 individuals, but they do not work for the 4th. And we can basically reduce the price of a single reaction. So we're able to do that. And then we use that knowledge for population genetics of both beetle and the pathogen. We completed that. And in the meantime, we completed that on a basically United States and Italian level regions. And we found something really interesting. So I'm not going to bore you with population genetics, but what is really interesting is that we have 2 different genetic clusters. So they're represented in 2 different colors for the pie chart. So in population genetics, what we usually considered original population is population that has a very high genetic diversity. And then the idea is the newly established population, like what we believed Italian population, should be relatively low genetic diversity. And that's not what we found. So that was extremely surprising. And we also believe that there's a multiple introductions from multiple sources. That is one of the hypotheses, or that walnut twig beetle was there. It was not damaging necessarily, but there was some evolutionary switch in the pathogen. We do not know what triggered that. So there are a lot of interesting ideas, hypotheses, and ways that we can explain this. But the bottom line is it is very genetically diverse. So it's either introduced multiple times or it has been undetected for a while. So another issue with TCD is detection. The pathogen G. smithia morbida is very hard to grow. So when we receive samples, what we have to do is peel the bark, find the galleries. And again, Eastern and Western isolates differ tremendously. It took us a while to obtain pure cultures from the East versus it took us a month or so, very easy process of finding galleries in G. smithia in Western isolates. Then we look under the microscope, then we basically create petri dish environment that more likely would be mimicking the wood itself. So these are moist papers, and then 3 to 5 days, usually these wood chips would have sporulation of G. smithia. And then what we can do, ideally, if we have something like this is a conidiophore, that's this fungal structure bearing conidia, we can basically pluck one of those. It looks almost like a bouquet, really. And then what we do is we do almost like very similar to bacterial streaking. And this is a fungus, this is not commonly done. But collaborator from Colorado suggested to do that because that way you will be highly likely to obtain pure culture and get rid of certain contaminants along the way as you're streaking. So 4 to 6 weeks later, if you're successful, this is what you get. You get a pure G. smithia culture. So the reality is it does not work that way. This is more likely what we see, a lot of contaminants, a lot of faster growing fungi that outcompete G. smithia. And this is very time consuming and laborious process on our end. Another complication with this is that what you see here on top are all G. smithia morbida isolates, and you can see morphological plasticity or morphological diversity of these isolates. They have been confirmed using sequencing. They're all G. smithia, but they look slightly different for untrained person. Also, the media that we grow that creates slightly different morphological colony formation. And if you're not familiar with G. smithia, other lookalike pathogens under the microscope can be very confusing. So confirmation process is very interesting and laborious, really. So what we were trying to do is to figure out a better way of doing that. So now that we have a species-specific markers, what that really means is that we can isolate DNA from this piece of wood. And if this was drilled through one of these holes, or if you peel that and you find a gallery and you drill directly through the gallery, the idea is because we have species-specific markers, we can isolate DNA from our target organism, which in this particular case was either G. smithia morbida, the fungus, or the vector walnut twig beetle. So we had a student, my master's student, ML, she worked in this and we published this work, and she became a complete drilling ninja. So she had some mad skills, but we were obtaining this basically drilled shaving from infested logs and isolating DNA from that, multiplying the DNA, and then visualizing this eventually using electrophoresis to see if we have presence of beetle or the pathogen. This is just to show you and summarize the differences that we're seeing between, at that time we worked with California samples and samples in Tennessee. So this would be our Western versus Eastern United States isolates. So ML was successful in obtaining clear peaks. So with a peak like this, we would say we're confident that we have presence of G. smithia morbida in this particular sample, and we were confident that we have presence of walnut twig beetle. We designed these markers to be far away from one another, so there is no confusion, but also we designed them with the idea that we can probably and hopefully put them together in a single reaction and reduce the cost a little bit further. So this worked pretty decently. For the state of Tennessee, we got the wood from 40 different trees, but not every single tree was confirmed that it had disease. It was just from the infested county. For California, where we had higher confirmation rate, we got it from infested orchard, and more likely all trees were infested. So this is something, and then our control was Missouri, which is the disease-free state. Still, we were trying to figure out if we can use basically our tools to detect presence or absence of the pathogen or the beetle in these samples. This system is good and works well. However, it requires, it's still around $10 per sample, and it requires specialized equipment. So what we wanted to do is, so we know it works, and we had another student and the Forest Service grant to expand that idea and use the basically qPCR, which is the gold standard for confirmation, just as a method that works for sure. But we wanted to simplify this and make it usable so people at disease clinics across the country could utilize this as a methodology, and very simple, going basically from several weeks to 8 to 10 hours to confirm the presence of the pathogen. So we wanted to take this and just do a very simple gel, which any lab with PCR machine could do. And it does not require extremely specialized equipment. And then the third option was basically to use a very simple color change of the tube if the pathogen is present or not, putting the specialized glasses. So we're able to use that and confirm that, yes, we can do that. And we use the gold standard to confirm that both of these methods work. And this was also published recently in Plant Disease. So we have several systems and several methods, and we are not the only group that did this. There were a number of different groups that developed really interesting approaches, including more recently a group from Italy. They developed a LAMP protocol. So this improved diagnostic is basically using the TaqMan probes, and I'm not going to go into details, but it's really, the idea is to simplify the system and use basically visualization through the light blocking glasses. And confirm the presence or absence of infested sample. So because we're dealing with this in Tennessee, and there are some states around us that don't have it, but the confirmation of the beetle and the pathogen is pretty complex, we wanted to explore several different options. So we wanted to see if we can culture basically or wash off and sub different fungi from the insects that are collected in these galleries. So we had a student who tried to do this, and the first method did not yield a single Geosmithia culture after doing over 2,000 washes. And then we realized, okay, this is what research is all about. So we need to move to option B. So option B was basically to utilize our molecular tools to homogenize, or basically take the whole beetle that we identified with whatever hopefully Geosmithia cultures it carries, and then extract DNA and use our Geosmithia specific marker to confirm presence or absence of Geosmithia in those samples, because he was unable to culture any of that. So we found something really interesting. So this work was done by Karan and Ramina, the postdoc in my lab at the time, and she's running disease clinic now in California, in Florida. And so we found basically 11 alternative vectors and really no vector specificity among these different Geosmithia species. So that was something interesting, something that we did not expect. But also we found that although these 11 vectors are important or maybe play a role, this was only done for East Tennessee. We haven't done this countrywide. And then we do not know their role necessarily in transmission of the disease. We also wanted to do pathogenicity, but and figure out the virulence of some of these pathogens, isolates of Geosmithia, and if they can induce, because we found a lot of genetic diversity. So this is a asexual reproducing pathogen. So we're trying to figure out why do we have such a high genetic diversity and very different infection rates in the East and West. So to do that and to control for genotype, we collected from 2 disease-free trees. We use a detached BALT assay as a proxy for virulence. We inoculated that, measured the cankers, and found something really interesting. We selected isolates that came from different genetic clusters based on our, so cluster 1 would be in purple and cluster 2 would be in orange. We found that basically there is a genetic difference between isolates in these different clusters, and then the canker size differ depending where these isolates are from. So there is a difference between the cluster themselves, but it's not necessarily based on East or West, which is what we originally thought. So that also confirms the idea of multiple introductions from multiple sources. So this is definitely human-mediated movement, and we're trying to figure out options of going a little bit further. So in collaboration with Purdue, we started working on a number of entomopathogens, or basically pathogens that could be controlling or be used as a biological control for the walnut twig beetle. And we called this particular experiment a squirrel experiment. Because again, there are a lot of failed things in research. So what we did is we soaked in Bavaria, trying to use that as a soaking the seeds and then eventually planting them and seeing if there is some kind of a systemic control that we can use for entomopathogens. But what we failed to do is to cover these pots, and then squirrels ate every single seed that we had. So we called it Squirrel Project from now on. In the meantime,
Janita, you have about 5 minutes.
Okay, thank you. In the meantime, also we work in collaboration with Purdue and HDRC on a long-term project we have in Oak Ridge near UT, where we planted basically walnut families, walnut trees from different families, from different breeding programs, and we'll eventually test them for a resistance to TCD. This is a at least 10-year project. So we have a lot of work in a pipeline, but this is something that spiked our interest in biological control. So what we found when we tested isolates or trees, infested trees, to see what is the fungal diversity of individuals from Tennessee and California, we found that a lot of Eastern trees basically contain trichoderma, which is also used as a biological control. But we did not find a huge amount of trichoderma in California, which could explain some of the differences that we're seeing between East and West. And then this sparked another set of projects related to control strategies. So this is a dual plate assay. This is with the initial idea to plant basically to place Geosmithia in a petri dish, and then a trichoderma species, which is the biological control. And if the reaction is compatible, you can see that trichoderma will eventually overgrow Geosmithia if the reaction is compatible. So we see this as a native, as a resistance in native trees across the Eastern United States in general, and that could explain some of the differences we're seeing. In 2018, there was a first chemical control, phosphajet, that was actually labeled for treating walnuts. And we played with that to some extent. And this is the basically PhD project of my student, Aaron Onofrak. So he did basal spray and then the drench, basal spray with phosphajet, and the drench experiment with trichoderma, trying to figure out if that can be used as a biological and chemical control for Geosmithia. So we treated the trees, and then several days post-treatment, we inoculated trees with Geosmithia. And then we did a number of transcriptomics that's really trying to figure out how the tree responds and what genes it turn on and off when the pathogen is present, and then eventually comparative genomics as well. I'm going to pass the microbiome work that I wanted to discuss. But beyond TCD, I wanted to also say that we're using that system, the complex system that involves host pathogen and the vector, to work on two other similar systems. And this is where collaboration with Ellen came in. One of them is the laurel wilt. And we are working on molecular detection of that. And the interesting part with laurel wilt, because of the climate change, because of these are the projected rate of laurel wilt. This was supposed to be by 2045 that we're seeing that into Texas and not coming to Tennessee at all, but we're finding laurel wilt in Tennessee and Kentucky. So there are changes, and we're trying to figure out a little bit better molecular detection method for that. And using, again, TCD as a placement, we're also working with oak wilt and the same idea, molecular detection, virulence, and host responses as a result of disease pressures. So I'm going to wrap it here and say that everything I presented here is really a work of enormous army of people, students, postdocs, technicians, collaborators, and this is my lab, and I work closely in collaboration with Bill Klingeman, Plant Sciences, University of Tennessee. He's my entomologist and man who shuts down my ideas when they're not that great. Fantastic collaborator, and this is my info. So I'd be happy to take any questions if you have.
Wonderful. Thank you so much for that presentation and all the work that you're doing with Thousand Cankers disease. And please go ahead and put your questions in the chat if you have them. I will also post the ISA CEU code. That's going to be KY22034. Again, that's KY22034. But I have a question to start us off, and that is, maybe you could expand a little bit on this. When Thousand Cankers disease was first detected in walnut, we care so much about walnut, right? It's an important species. It's valuable in our woods as well as in the urban landscape. I think a lot of people were really concerned about it. But it hasn't really been as big of a problem in Kentucky, at least. We haven't detected it. It hasn't spread here or in other states in the eastern US. So kind of what do you see as the future for Thousand Cankers disease in our area? Is it something that people should still be concerned about? Is it maybe lower on the priority list now relative to some other things? Kind of what's your synthesis of that?
So I have a love-hate relationship with that. So it's not just because we work on that, but to answer some of the deeper questions, to understand the biology, genetics of the host, the pathogen. So this is a complex system. So to understand any of that, you require years of research. So we are right now 10 years or plus later to answer some phenomenal questions that actually could be translated into similar complex systems like that are fungal vector by a beetle or some kind of insect that could be used as potentially model organisms for future research. And I know we have to shift towards the more emerging threats and issues, but it's almost shameful not to continue something like this. And I think the reasons we're seeing this is one of the explanations could be the trichoderma and the natural resistance in our native trees here. So we have a potential to find trees that are potentially resistant, and maybe that's why we're not seeing this as much. So if it spreads worldwide, we may be the first to have a solution how to create resistant varieties or breed for resistant or something along the lines, because breeding programs do require quite a bit of time. So it's much more complex issue. And I think another problem is we detected it a few years after following the very severe drought that we had here. And there is anecdotal evidence that the drought does play a very significant role, but we do not know that for sure. So we actually, Aaron is doing the microbiome that's drought induced. So we're going to get some answers, hopefully. But that can be also another option that we are using for other trees that are maybe recovering now, but another drought or another issue of that sort can create, I don't want to say reinfestation, but versions of that because the trees are not strong enough to respond to that much disease pressure. That was a long answer too.
No, I think those are some really good points, both in terms of maybe why it hasn't panned out to be as destructive as initially thought, but the importance of continuing to be on the lookout for thousand cankers disease as well as other issues, and the fact that the current situation might change in the future and might change as it arrives to other locations. So thanks for that. We did have one question in the chat about, as far as vectors go, have other insects like hemipterans been found to be able to vector it, or are the only known vectors beetles?
So I do believe they're beetles so far, but that would be more Bill question than me. So what we found, so this is why I don't feel extremely comfortable to say we did find 11 vectors, but what is their role with Geosmithia morbida is still to be determined. And this was only very small geographical range, which is Eastern Tennessee, so which can be very different based on the region. That is something I would love. It's a very hard project to do, but that is something I think it's needed, and it would be really nice to explore. Because I think it's going to be more present and we're going to have more vectors, but understanding and depicting the role would be a little bit harder project to wrap my mind around.
Excellent. Well, thank you so much. We appreciate you talking with our group today and look forward to hearing more about, unfortunately, no, I shouldn't say look forward. We are not looking forward to hearing more about diseases in our area and their complex vector interactions. So thanks again, and appreciate you providing your information there for anyone else who might have other questions.